Hey folks, it's Ethan Paulaner, co-founder of Cultra, the nutrient analytics platform for agronomists and growers. In this video, we'll be exploring the role that earthworms play in healthy soil environments. Let's dive into it. Earthworms represent one of the most consequential yet overlooked organisms in soil ecosystems. These segmented oligochaetes vary dramatically in scale, from a few centimeters to over a yard in length, with over 7,000 species distributed across both agricultural landscapes and residential gardens alike. In thriving systems, populations can reach densities of 10 to 50 earthworms per square foot, translating to approximately 3 million earthworms per acre. Operating at this magnitude, they displace nearly 18 tons of soil annually during foraging activities, functioning as persistent soil engineers that fundamentally reshape substrate architecture. To grasp their ecological influence, we'll examine their reproductive strategies, feeding mechanisms, and their cascading effects on soil structure and nutrient cycling. Understanding earthworm influence begins with their reproductive capacity. Though hermaphroditic, meaning they have both male and female sex organs, earthworms require cross-fertilization. Following mating, each individual secretes a mucus band that hardens into a cocoon that contains roughly 15 or so embryos. These embryos reach sexual maturity within 3 to 4 months, and with certain species achieving lifespans of 15 years, we can consider this reproduction as a perpetual production system. Once established, it sustains itself across multiple generations, continuously replenishing soil engineers. This accounts for the rapid population expansion observed under favorable conditions. Earthworms lack both eyes and teeth. Instead, they detect light through photosensitive cells on their epidermis and manipulate food using their prostomium, a muscular sensory lobe that overhangs the mouth. Their diet consists primarily of bacteria, supplemented by fungal hyphae, protozoa, nematodes, and decomposing organic matter. Ingestion begins when the muscular pharynx creates negative pressure to draw material inward. Food progresses to the crop for temporary storage before entering the gizzard, essentially a biological grinding chamber. Sand grains and mineral fragments within the gizzard pulverize organic matter into fine particles. Since earthworms synthesize no digestive enzymes, this mechanical reduction is critical. Symbiotic bacteria within the intestinal tract decompose this material, liberating nutrients for absorption. Unassimilated residues are excreted as vermin castings naturally, enriched with calcium carbonate, providing modest alkalizing effects. The earthworm's digestive system operates as a biochemical processing facility, transforming restricted substrates into bioavailable compounds. And this metabolic conversion establishes the foundation for understanding vermin casting chemistry. With digestive processes established, we can address nutrient transformation and an often overlooked benefit, pathogen regulation. Vermin castings contain approximately 50% more organic matter than surrounding soil, substantially increasing cation exchange capacity, which is the soil's ability to retain nutrient ions. More significantly, biochemical activity during gut passage mobilizes nutrients typically sequestered in stable organic complexes. Now, relative to unprocessed soil, vermin castings may contain up to 7-fold increases in phosphate, 10-fold in potassium, 5-fold in nitrogen, 3-fold in magnesium, and 1.5-fold in calcium. Across 1 acre, earthworms may deposit 10 to 15 tons of castings annually, effectively operating as a continuous nutrient mineralization system beneath the soil surface. Now, beyond fertility enhancement, vermin castings demonstrate remarkable disease suppressive properties. The passage through the earthworm gut creates an environment hostile to many plant pathogens while promoting beneficial microorganisms. And research consistently shows that vermin casting suppresses soil-borne diseases and various root rot organisms. Multiple mechanisms drive this suppression: competitive exclusion by beneficial bacteria, production of antimicrobial compounds during digestion, and enhanced populations of predatory microorganisms that consume pathogens. In essence, earthworms curate the soil microbiome, favoring organisms that protect rather than threaten plant health. Think of earthworms as a biochemical catalyst that simultaneously reorganize nutrient pools and restructure microbial communities, converting complex, unavailable compounds into plant-accessible forms while suppressing disease. Beyond nutrient cycling, earthworms profoundly influence physical soil properties. As primary detritivores in the soil food web, they fragment leaf litter, increasing surface area for microbial consumption of cellulose and lignin. In systems with robust earthworm populations, leaf litter requiring 18 to 24 months of decomposition can fully mineralize within just 3 months. Now, their burrowing activities generate extensive channel networks that enhance aeration and water transport. These galleries subsequently fill with casting material and organic fragments, creating nutrient-enriched corridors that roots preferentially colonize. Certain species evacuate vertical shafts extending 12 feet, meaning they travel up and down. Others translocate surface organic matter to depth, while others maintain horizontal galleries near the soil's surface, traveling on the x-axis. Now, earthworms are capable of displacing rocks exceeding 6 times their body mass, demonstrating their remarkable geophysical influence. The hydrological impact of these burrow networks cannot be overstated. Earthworm channels function as preferential flow paths, increasing water filtration by up to 10 times compared to compacted or worm-free soils. Now, during intense rainfall events, these micropores rapidly conduct water deep into the soil zone, reducing surface runoff and erosion while recharging groundwater reserves. Earthworms themselves require consistent moisture. They thrive when soil content maintains 60 to 80% of field capacity. Now, below this threshold, many species enter dormancy, coiling within mucus-lined chambers until moisture returns. Now, consider their burrow systems as biological infrastructure, channels that regulate gas exchange, water infiltration, root exploration, while also mitigating surface runoff during precipitation events. This infrastructure simultaneously creates the moisture conditions earthworms themselves require for sustained activity. Now, consider their burrow systems as biological infrastructure, channels that regulate gas exchange, water infiltration, and root exploration, all while mitigating surface runoff during precipitation events. And simultaneously, this infrastructure creates the moisture conditions earthworms themselves require for sustained activity. Understanding earthworm ecology requires acknowledging mortality factors. Natural predators, primarily birds and small mammals, exert limited population control while contributing microbial inocula and nutrients through excreting fecal matter. Human disturbances pose far greater threats. Mechanical tillage and soil inversion cause catastrophic physical trauma, and most earthworms cannot regenerate from bisection. Salt index fertilizers can create osmotic stress and chemical irritation, precipitating dramatic population declines. Conversely, when mechanical disturbance ceases and chemical inputs moderate, populations typically recover pretty rapidly. Earthworms fragment organic residues, engineer soil structure, and dramatically increase water infiltration and retention capacity. They enrich substrates through casting deposition, suppress soil-borne plant diseases, and accelerate decomposition processes. Their activities sustain diverse microbial communities and maintain nutrient-dense, resilient soil systems. Abundant earthworm populations serve as a reliable bioindicator of soil health and functional ecosystem integrity. And by comprehending their reproductive ecology, trophic relationships, hydrological influence, and environmental sensitivities, we develop evidence-based strategies for cultivating biologically active, inherently fertile soils. I'm Ethan Paulaner, co-founder of Cultra, a nutrient analytics platform for agronomists and growers. If you're interested in learning more about your soil biology and chemistry, please take a look at our other videos. If you're curious to learn more about the tool, visit our website. Thanks so much. We'll see you in the next one.